Okay, thank you, Becca. Good morning, everyone. We're going to be presenting on a project related to invasive carp invasion in the Ohio River. And I'm going to pass it very shortly to Aaron Shepa and Luca Marquez to present their work, the PhD student and master's student, respectively. We're in the final stretches of the project. We believe our funding is going to go until June, July of 2025. And so our project's much like the story of Abe Lincoln, right? And when he was asked to cut down a tree in an hour, he said he'd sharpen his axe for 45 minutes, right? Well, we've been sharpening the axe, gathering data, developing models, and the fruits of that labor are wood chips flying about now, right? So we're in the final parts of the project, but dominoes are going to fall really quickly. So Erin's going to lead off, talk about the model she developed, a simulation, which basically validates the model. And then from there, the actual applications are going to move fast. So before I pass it to them, I also want to acknowledge that our project was originally designed for the Ohio River and Tennessee Cumberland River. We met some, not necessarily resistance, but we weren't able to gather data for Tennessee Cumberland, and early on made the decision, rather than slow down, we're going to push forward with the Ohio River data. So we're focusing there, and my hope is once we have products of the Ohio River that other watersheds are going to want to adopt those. And I'm a member of the data analysis work group for the ICAC, which is the invasive carp advisory committee. So, hopefully be able to help disseminate the products from this project to other partners. And with that, then I want to acknowledge Katie Zipfel from West Virginia DNR, who supplied data, Andrew Stump, Jeff Hayrod, Chris Hickey, and Maris White from Kentucky DNR shared data, as well as Craig Jansen from Indiana DNR, and Ryan Arlo and his team from Arsenco. So that data basically what went to Aaron and Luca for their projects. Questions at the end? Carol Rontas, the co-PI of the project, she's teaching right now, so couldn't be here, and I'm going to have to drop off because I teach at 11. But Aaron and Luca are very well equipped to answer any questions on the project. With that, I'll pass it to Aaron.
I apologize. I forgot to unmute myself. Okay. Well, with that, I will start with my portion of this presentation. So like Brent said, my name is Aaron Shepta. I'm a doctoral candidate, and today I'm going to be sharing some preliminary results from a model that we developed to try and understand factors that may be limiting the establishment success of invasive bighead and silver carp in the Ohio River Basin. So there are 4 species of carp that are considered invasive in the United States. The ones that we focus on here are the bighead carp and our silver carp. Both of these species are native to Eastern Asia, and they've been introduced to many countries around the world, with the bighead carp being introduced to 70 different countries, including the United States, and the silver carp being introduced to 88, including the United States. Both of these species are associated with severe ecological and economic impacts in systems where they've been introduced. Both of them are large-bodied, low-trophic planktivores, which means that they feed very low on the food web, and they're able to consume a lot of biomass that would otherwise be available for juvenile fish and other planktivores in the system. Economically, this means that they have the ability to disrupt native fisheries, but they also have this interesting economic impact of potentially injuring boaters and recreational fishers as well. So you can see here, the thing about silver carp that makes them pretty dangerous is they actually tend to jump when they get frightened. So this is a problem for not just our recreational anglers, but anybody who wants to go in the Ohio River and go on a boat, as they risk taking a 30-pound carp to the face which can be very detrimental in these systems. So some background on their introduction in the United States. Those species were first introduced in the lower Mississippi River Basin in the 1970s and have since spread and expanded their range within the United States. So when they reached the upper Mississippi River Basin, their populations took off and they spread very rapidly. Conversely, in the Ohio River, where we do our research, their spread slowed and even stalled in some locations. So for our research, the question then became why? What is it about the Ohio River Basin itself that might be impeding their spread? And can we apply lessons that we learn here to help manage their spread in these other basins as well? So for us, like I said, we're focusing on the Ohio River Basin. Managers typically break this river up into a series of what they call invasion fronts. And these fronts really just describe a gradient of carp abundance, reproduction, and recruitment. So if you see down here in our red, we think that carp are highly abundant, adult carp, and they're actively reproducing and recruiting. But as we move upstream in the Ohio, in this yellow area, we kind of lose that recruitment aspect. And then when we move to our green, we lose reproduction pretty much altogether. And then upstream in our blue, we think that there's no adult carp that have ever been found. While this is what we consider our invasion fronts, currently there's no true measurement of recruitment and reproduction success across the Ohio River Basin. So that's kind of where our project here comes in. We wanted a good measurement of both reproduction and recruitment success of these species in this river. So to do this, we sampled carp across 3 different age classes, the first being eggs and larvae. Then we also sampled for juvenile carp, which are sexually immature individuals. And finally, we sampled for adult carp. Each of these different age classes was sampled using a variety of different gears. For our eggs and larvae, we mostly did these ichthyoplankton trawls. For juvenile carp and adult carp, there was either electrofishing and different kinds of gill nets, pike nets, a lot of different effort to collect these fish because, as you remember, they can jump, so they're incredibly difficult to sample for. We had an interesting, I guess I'll say, problem with the data that we had, where it was sampled in such a way where we didn't necessarily have the best method to analyze it. So I wanted to explain a little bit about our sampling design, the way that this data was collected. It was kind of complex spatially and temporally. So the Ohio River Basin, or the Ohio River, can be broken up into a series of pools, with the pool being the area between 2 dams. These are incredibly long stretches of river. They span between 41 and 114 miles long. And researchers went out and they sampled these pools on a yearly basis for these different age classes. Nested within each of these pools, however, they also visited a series of sampling sites. Each of these sites was sampled on a yearly basis. However, there was also several visits paid to each of these sites within a year, during which researchers took replicate detection, non-detection surveys. So this is an incredibly complex nested sampling design, and it opened up for us to kind of extend our traditional occupancy modeling techniques into something that we're calling a dynamic multi-scale occupancy model. So that sounds very fancy, but essentially the things that we wanted to look at with this model is at that large scale pool level, we wanted to see yearly colonization and persistence probability of these carp. Also nested within each pool, we wanted to look at colonization probability and persistence probability for each year for each site. And finally, we wanted to look at those colonization and persistence between those visits within a year. And we could lastly use these replicate non-detection detection surveys to look at detection probability. So this is a lot of parameters, but essentially these large scale parameters, we really wanted to see which of these large stretches of river are we seeing this constant recruitment and reproduction. For our small scale parameters, we wanted to see sites within these pools that are likely supporting our carp populations and our successful recruitment. And then for our intraannual populations, we really wanted to see our adult carp, where are they throughout the year? And if we find juvenile carp, is it true recruitment? Are they there all year? So because this is a new kind of modeling technique, what we needed to do was we needed to simulate some data and validate the model structure to ensure that it was valid and it could make predictions across a range of different sampling designs. I won't get too much in the weeds of what we did, but essentially to simulate data, you specify different kinds of input parameters, you specify different sampling designs, you tell the computer to simulate data, you run these models, we did it 1,000 times each, and you assess the output. So I have some preliminary results from this to show that our model was valid. So essentially what we did, we had this base model here of 16 pools, 4 sites, 4 years, 4 visits. And we wanted to know if we had less pools, how valid was our model, more pools, how valid was our model, and the same with these different spatial and temporal replicates. Could this model handle these different levels of replication and still give us good outputs? So first, when we were looking at our large-scale sampling unit, here on this graph to orient you, we have our different model parameters that we were estimating, and we have our probability. I did an input of 50% of 0.5 probability, so that's this dashed line. So that's our real parameter that we used. These dots, when they're closer to that 50%, they're more accurate. And then these error bars, you would expect them to become more and more precise, so smaller, as we increase our replications. So this is what we're talking about here. Accuracy, the dots are closer to the 50%. Precision, our confidence intervals are smaller, just to orient you guys. So when we change the number of pools, as we see an increasing number of those large scale sampling units, we did see an increase of accuracy across all the parameters and an increase of precision. When we looked at the sites nested within those pools, again, we saw an increase is correlated with an increase in accuracy across all these parameters, and we saw increase in precision with our small scale annual and intraannual parameters. And we did see an increase in precision of our large scale parameters, only up into a point where it kind of stopped here around 8 sampling sites nested within would increase that precision. Temporally, when we change the number of years, again, we see that all parameters were more accurate, and we saw an increase in precision in most of our different large-scale, small-scale parameters, except for our initial occupancy, which isn't necessarily a problem here. It's what we would have expected. And finally, looking at our number of visits, again, we see an increase in accuracy in mostly our small-scale parameters, and we see an increase in precision of most of our small-scale parameters as well. So basically, what does this mean for our carp data? This is just a lot of work to make sure the model actually worked. Essentially, what we're looking at, for our adults, we had a lot of sampling effort for our adults. And it seems like this model is going to run really, really well and give us very accurate and precise readings. And same for our juveniles. The only thing that I would be worried about with our eggs and larvae is we might want to suggest increasing this number of years, because what we saw with our model output is if you increase those number of years, we'll probably get better precision with some of our estimates for our eggs and larvae. In the end, we want to use this data set to predict our different occupancy for our adults, our juveniles, and our eggs and larvae. And we're going to use this data in combination with results from my research, Luca's research, and other collaborating researchers to identify priority locations for management of carp on the Ohio River, to potentially stop their spread. So that's where this all is going in the future. And I just would like to briefly acknowledge all of our different funding sources and our collaborators. And I don't have that much time, so I will say thank you here. There's my email if you have questions, and I will quickly pass it over to Luca for his portion of this research. Thank you.
Hello, my name is Luca Marques do Amaral. I'm an international graduate student, co-advised by Dr. Francis and Dr. Murray, and I'm working towards my master's degree. It's titled Assessing Ecological Impacts of Invasive Carp in the Ohio River. So, as Dr. Murray and Aaron said, we know that invasive carp have been introduced and they haven't been able to invade the entirety of the Ohio River. And although we do not know why, it is also important to understand how they could be affecting native fish, right? Biologically speaking, we know that impacts may vary spatially, temporally, and between the different assemblages or aggregations of different species of fish throughout the river. Then my objective is basically to gain insight into potential impacts that invasive carp may be causing to those native fish across space and time, and think of potential mechanisms that could be behind their invasiveness. Can you next, please? Twice. Sorry about that. So we need to think about how carp impacted other ecosystems. We have two interesting examples. One is in a lake in France. Basically, they experimented and they saw that silver carp provoke shifts in assemblages of phytoplankton and zooplankton, which are very important not only for energy but also for nutrients cycling. Zooplanktons are connecting the lower end of the trophic web to upper ends, so disruptions in those two levels may cause a disruption in the entire food web. So as Aaron said, um, they can disrupt um, food webs because of this. They can co-opt biomass, and not only that, but also nutrients and energy. Aglaton did an also interesting work here in the US. Basically, they're trying to understand if fish assemblages differed before and after big head carp establishment, and they also observed a shift in native fish assemblages. So how do we study? Can you please, thank you. So how do we study this? So Egerton used the taxonomic approach, which is basically a traditional approach on species diversity. So what is the number of the species, um, um, measures such as that. And several studies have been using other facets of biodiversity, such as functional and phylogenetic. So basically, um, functional works with the unique what we call traits, which are basically characteristics of species um, that can affect their survival, growth, and reproduction. And phylogenetic are just a kinship, right, between species. And we can use those different components of biodiversity to have a better understanding of the overall picture and understand the impacts. Next, please. Thank you. So then how, what data are we using, right, to accomplish that? I'm using the Ohio River Valley Water Sanitation Commission, or SANCO. They have fish count data that is publicly available online. Next, please. And I'm also using functional traits data set published for freshwater fish from two data sets from Ross and from Frimpong. Next, please. Here, as Aaron said, data can be challenging sometimes. So this is basically a map of all the points collected for the Orsenko data set, I'm using from 91 to 2023, which is a span of 32 years. Basically, they collect 2 to 4 pools every year out of the 19 navigational pools because of um, constraints such as time and resources. They collect those by boat electrofishing, and I've been working on this data and try to make it ready for analysis for a while now. And we came to a number of more or less 48 species of native fish, excluding the 3 carp that they found: grass carp, silver carp, and big head. They didn't catch any black carp, potentially because of their sampling method. Next, please. So how are we gonna analyze this data? We have our question, right? How 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 they're impacting those native fish assemblages. So I'm gonna use a hierarchical modeling of species community, which can account for the temporal and spatial autocorrelation that Aaron also talked about. Places are close, and that affects values of several metrics. And basically what we have is a community matrix where we have our sites and our species, and I'm gonna try to explain the variation that carp causes on those native species using some variables such as the year, the coordinates, maybe temperature, and of course carp. Next, please. So for our expected results, we expect that invasive carp are altering native fish taxonomic composition over time and across space. For example, when some there a decline in species richness, are there less species since carp came? They're also provoking shifts in functional traits. So what are the unique characteristics of species where carp invaded? Did they change across time and space? And two specific predictions is that invasive carp are provoking declines in native planktivorous fish because they would be um, competing with them, and also that they're provoking declines in native predator fish, because if carp is competing with planktivorous fish, um, planktivorous fish would have a decline in abundance or presence, and that would affect predator fish that depend on those. For management implications, so basically what that means is we can understand what kind of communities are susceptible. Are planktivorous predominant, communities susceptible, piscivorous predominant, both are impacted, expected to be severe, or impacts expected to be mild. What are the possible mechanisms that could be behind this, right? I mentioned competition, but there are so many possible impacts. And by having those informations, we can inform better management of those native fish based on those vulnerable groups. Next, please. So that's all. I'm sorry it was a little rushed. We don't have a lot of time, but I just wanted to thank you all for listening to me, West Virginia University, West Virginia Water Research Institute, USGS, and Orsenko, and all of you. Thank you.
All right. Thank you so much, Aaron, Brent, and Luca. We are on to our second presentation with Leslie and Citraka. So if you two could introduce yourselves, and we're excited to hear more.
Good morning. Happy to be here and talk with you all about our project, long-term water quality monitoring in the Monongahela River Basin, and introduce our group, Citraka Rabaman-Kasoa and Sarah Nelson. There's 2 students. Sarah's an undergrad student. She did the initial data analysis for this work. Citraka continued and extended to the great amount of statistical tests that we'll present today, our results. And then Rachel Spiernack and Melissa O'Neill with the Water Research Institute and their great knowledge of the Mon River Basin and the Three Rivers Quest program, which provided the backbone and all of the data for this project. So we'll just share what we learned. Okay. So this is our setting. We are focusing in on the Monongahela River Basin, highly industrialized, and includes the West Virginia and Pennsylvania coal fields. Historically, acid mine drainage is the main water quality concern. AMD originates from deep mine openings or surface mine pits at abandoned mine sites and is characterized by low pH and high concentrations of things like iron, manganese, aluminum, and sulfate. Traditional gas drilling, industrial and municipal pollution, and Marcellus shale gas development also contribute to water quality concerns in the watershed. And a little bit of history of the watershed, which resulted in this long-term data set that was available for us to analyze. And you may have been familiar with some of these things in history here. In 2008, concentrations of sulfate and total dissolved solids that exceeded the drinking water secondary maximum contaminant levels were observed in the Monongahela River. Historically, these high total dissolved solids concentrations were only observed at low flows. During this time, there was increased gas development in the region, and the relative contributions to this TDS load from the coal industry and the gas industry were unknown. So, as a result, motivated by these elevated levels of total dissolved solids, biweekly water quality monitoring of the Mon River and its major tributaries began in July 2009 by the West Virginia Water Institute. 4 sites on the Mon River and the mouth of 12 major tributaries were monitored, focusing on TDS and its major constituents. The sampling continues today as the long-term Three Rivers Quest monitoring and reporting program, and that's the data set that we're focusing on today. And to continue a bit of the history of the Mon River, in 2009, a massive fish kill and complete mussel kill occurred in Duckard Creek, major tributary to the Mon River. This event was triggered by a toxin as a bloom of algae, golden algae, resulting from high TDS levels. So then in December 2010, the Mon River was listed as impaired for sulfate by the West Virginia Department of Environmental Protection. Result from the initial water quality monitoring suggested that treated mine drainage were controlling factors to the TDS load. So using these monitoring data, West Virginia Water Research Institute developed a non-mandated total maximum daily load management plan to reduce those TDS levels. The approach was applied in January 2010 by the coal industry on a voluntary basis. In addition, Pennsylvania passed TDS regulations to address new or increased TDS resulting from natural gas production in 2010. Pennsylvania restricted flowback from hydraulic fracturing into publicly owned treatment works in May 2011, and in May 2013, a reverse osmosis plant began operations in Mannington, West Virginia to treat wastewater from the coal industry. Sulfate and TDS levels have met EPA standards in the Mon River since the application of these management strategies, and the Mon River was removed as impaired for sulfate in December 2014. So that's a long history of the watershed, but what resulted, that initial monitoring turned into the Three Rivers Quest program, and that resulted in a long-term data set of water quality monitoring data. And since then, the 3RQ project has expanded to monitor the Mon River, the Allegheny River, and Ohio watersheds. Rivers, tributaries, and headwater streams draining more than 60,000 square kilometers are monitored. Academic researchers, citizen scientists, and conservation groups collect, analyze, and monitor water quality, and the results are published for the public. So as a result of these efforts, a long-term water quality record for the Monongahela River Basin exists since 2009. So our task was to analyze that long-term data set and look for overall trends and what we've seen over time in the Monongahela River Basin. So our overall objective was to evaluate long-term trends in water quality in the Monongahela River Basin. So we use this publicly available 3RQ, Three Rivers Quest data set, and we applied a trend analysis approach and to determine if there were trends in water quality parameters of the Monongahela River and its major tributaries. Okay, so this is our setting. This is highlighting the Mon River Basin. The water quality data were collected as part of the Three Rivers Quest monitoring initiative, focusing here on only the Monongahela River Basin. It flows from north central West Virginia to southwestern Pennsylvania, and it ends at the confluence of the Allegheny River. And you can see the monitoring sites that were the focus of this study. We'll zoom in on those. So we used long-term records from 18 sampling sites. 6 were on the main stem, and they're identified. They're identified by river mile number on the main stem, and then 12 locations on major tributaries. The sampling locations were originally selected to determine the effects of acid mine drainage treatment plants, major cold fire power plants, and a brine treatment facility on water quality. So you can see the relative location of the sampling sites and where they were into relative location of these major treatment facilities, coal fire plants and brine treatment facility in the right figure. And also see the location of the major tributaries, and those arrows are scaled to the drainage area size. So we didn't collect the data as part of the project. We were using the long-term data set. This data continues to be collected, but we were using water samples that were collected and field samples that were collected beginning July 2009. Initially, they were collected at a 2-week interval, and then in 2015, the frequency was reduced to monthly. We focused our analysis only on data that were collected and recorded by the West Virginia Water Research Institute. We didn't include any data that were collected by watershed groups in this particular analysis. So at each site, a 500 milliliter unfiltered grab water sample was collected under the water surface, and a 250 milliliter water sample was filtered through 0.45 micron filter, and it was poured into a pre-acidified bottle. Those samples were collected, transported to the lab for analysis, and the samples were analyzed for alkalinity, sulfate, chloride, dissolved aluminum, calcium, iron, magnesium, manganese, and sodium. TDS was calculated as the sum of the concentrations of all measured dissolved constituents. Acidity was estimated. And then a YSI multi-parameter instrument was used to measure conductivity, pH, and temperature directly in the field. As part of our statistics, we also accounted for effects due to discharge. So mean daily discharge values were either obtained from nearby USGS gauge stations at the time of sampling, estimated with relationships reported by Kingsbury et al., or estimated by the Watershed Modeling and Characterization System. This table summarizes all the sampling locations, the contributing drainage area, and also in the final column lists the dates of available data, because some monitoring sites came on later in the monitoring program. They're added later. Some have small gaps of data due to funding availability, but this summarizes the locations of all of our monitoring sites that were used in the analysis. So really, this was a statistical analysis of those existing data, and we performed a relatively traditional trend analysis. We focused on non-parametric tests. We performed 4 non-parametric statistical tests to determine if there are significant significant monotonic trends over time at all of those monitoring stations. First, a man-kindle trend test. We also did a man-kindle trend test, which was adjusted for discharge, a seasonal kindle test where we define season as monthly, and a seasonal kindle test, again, season as monthly and adjusted for discharge. And we did all of our statistics using R. And this is a snapshot of our overall results, and we will focus in on what this means in a second. But this gives you an overall view of the trends and the complete system in which we did the analysis. The upper red arrows are a significant increasing trend. Downward triangles are significant decreasing trend, and the open squares signify no statistical trend was observed. It also summarizes all of the 4 tests that were completed at each site: the man-kindle test in the upper left, the seasonal man-kindle upper right, man-kindle adjusted bottom left, and seasonal man-kindle adjusted for discharge in the bottom right. This is also showing for all of the parameters that met the requirements for our statistical testing and all of the major monitoring stations. The top part of this table is showing trends in the main stem by river mile, and then the major tributaries in the bottom part of the table. But we'll zoom in on some of our more significant information that we found. So first, we did these 4 non-parametric tests to determine the presence of monotonic trends. Generally, all the tests resulted in the same results, regardless of whether the data were adjusted for discharge or considered for seasonality. Generally, we had the same either significant or no significant, significant increasing, significant decreasing, or no observed trend, regardless of test. And we're going to show you the results. The results here that I'm showing had significant, all in agreement, all 4 tests show decreasing trends when I'm showing an arrow in the next few slides. So first, we'll look at the main stem. In the main stem, decreasing trends over time were observed for levels of chloride, sodium, total dissolved solids, and sulfate from river mile 23 to river mile 102. And then for magnesium, we had significant decreasing trends for river mile 23 to river mile 89. And then in the main stem, we found decreasing trends were also observed for a portion of the main stem for conductivity, river mile 23 and river mile 82 to river mile 102, and for manganese, river mile 89. Levels of pH and alkalinity increased over time for a large portion of the main stem. For pH, river mile 11, 23, 82, and 102. And for alkalinity, river mile 23, 82, 89, and 102. Few significant trends were observed for temperature, acidity, iron, aluminum, and calcium are showing some of those significant trends which were in agreement for acidity, iron, and aluminum. Then the major tributaries, we found decreasing trends for conductivity, acidity, sulfate, iron, manganese, calcium, magnesium, chloride, sodium, and total dissolved solids in several locations, highlighting those here in the blue highlighted boxes. We saw increasing trends for pH, alkalinity, and aluminum for several locations, again highlighted in the boxes. And we found the fewest significant observed trends at 10 Mile Creek and Tigert Valley River. So we identify decreasing trends in sulfate, total dissolved solids among 83% of the main stem locations, regardless of statistical test. So this supporting the change of the impaired status of the Monongahela River. Similarly, total dissolved solids and sulfate decreased at 63% of all the monitored locations. Previous research suggests that TDS, chloride, and sulfate levels in the Mon River are seasonal and related to flow rate. most of the trends that we observed in the main stem were the same for total dissolved solids and chloride and sulfate, regardless of adjusting for discharge or seasonal effects. There has been other work by Kingsbury et al., Kingsbury et al. 2023, where it was using field estimates of total dissolved solids and evaluating trends for total dissolved solids in the Mon River. And our results support his those findings by that work. That work relied on field estimates of TDS. We relied on the sum of concentrations of all the measured dissolved constituents, and our findings still support this TDS trends in the watershed. Our study only evaluated gradual trends over time using a trend analysis approach. Only monotonic trends were evaluated. No step changes were evaluated for. Step changes may exist, but we didn't test for that type in this analysis. So overall, trends and acid mine drainage signals in the main stem either showed no trend or were improving. Of the 6 main stem sampling locations, widespread decreasing trends in total dissolved solids, sulfates, chloride, and sodium were observed, regardless of adjusting for discharge or season. Similarly, increasing trends or no trends in pH were observed at all sampling sites. Water quality gains are likely related to the voluntary management plan that was implemented by the coal industry. And I think that these results that we presented is important because this independent monitoring through the 3RQ program is important to communicate impacts as well as plan for future water management. So highlight partners that helped in this research. USGS supported the analysis and all of the support for the 3RQ program. And that's all into there. So thank you.
Thank you so much, Leslie and Citraka. We're going to do our last presentation. I'll have Hannah introduce herself in a second, and then we will have any questions from all of the presentations. So, Hannah?
Hi, good morning, everyone. Let's make sure I share my screen correctly. Cool. Maybe. There we go. Everyone's seeing it okay?
We can see your notes.
Oh no, I knew this would happen. Hold on. Let's see. Is it this one then? Maybe?
Yes.
Okay, cool. Okay, well, as Becca said, my name is Hannah Bentley, and I'm a water research and remediation student worker at the West Virginia Water Research Institute. And today I'm going to be presenting on the impact of drought on water quality in the Monongahela River. Well, oh, there we go. Sorry about that. So first of all, I want to talk about, what is a drought? A drought is the prolonged period of abnormally low rainfall that results in a shortage of water. Droughts fall under 4 different categories. So a meteorological drought is is the most common form of drought, and it's a period of below average precipitation. Then we have agricultural droughts, and those are the lack of precipitation that negatively affects crop production. We also have hydrological droughts, and those are the reduced water levels in water reservoirs such as lakes, rivers, streams. And lastly, we have socioeconomic drought, which is the water scarcity that impacts society and the economy. So droughts in West Virginia have been far more common recently. The Mon River in particular is important for drinking water and recreation, and as well as wildlife habitat. And so as these droughts become more common, we have to pay attention to them more. So I'm sure, as you all are aware, in the summer and fall period of this year, we experienced one of the worst droughts in about 25 years. And this was known as the driest 18 months since 1987. And it really put a strain on our water supplies and ecosystems. The decreased stream flow that was caused by these droughts really increased the concentration of pollutants, because there is a decrease in water and the water can't dilute those pollutants as much as it used to be able to. And this drought overall affected about 1.9 million residents across 51 counties. So the graph here is showing October. And as you can see, like, the middle of the state is really seeing that D4 level of exceptional drought. And then the entire state was experiencing some kind of level of drought. And then currently, the drought.gov monitor is showing that we are still in a drought, but it's not nearly as extreme, and it's more of the D0 through D2 ratings, but all of the state is still experiencing that drought. So impacts of drought. As I was talking about, climate change is going to be one of the largest impacts, and it's going to drive those more frequent and severe drought events. West Virginia as a state is predicted to have more rain in the winter and spring, and we're not going to see as much precipitation in the form of snow as temperatures increase. So this picture to the right is actually from July 2024, and it's showing a dried out branch of the Cheat River near Albright, West Virginia. And as I said, like, as climate continues to change, we're going to see shifts in rain patterns and the range with which these rain events fall under, and it's going to continue to contribute to flood and drought extremes. So we're going to see increased events of both. So 3RQ really monitors the water quality here. We conduct monthly sampling along the Mon and its tributaries, and we're really looking at TDS, which is total dissolved solids, and that's going to be the concentration of dissolved minerals, salts, and organic materials. And higher TDS levels are going to indicate impairment of those waterways. We're also looking at pH, which we use to measure the acidity and alkalinity of the waterway. And then we're also monitoring for heavy metals, primarily iron and aluminum, and those become more concentrated during drought events. We're also going to be looking at stream flow. So in 2024, that drought, we saw a sharp decline in flow levels. And then, as I was talking about earlier, decreased flow increases the concentration of TDS, and that impacts us and wildlife habitats alike. So we created a drought threshold, and a drought threshold is the point at which drought has reached a critical stage, and it's really just the point at which an event can be considered a drought. And so it's usually based on the temperature, soil moisture, precipitation, and stream flow. And then it's really important to understand the drought threshold because it's a proactive water management assessment, and we can use it to further predict times that are going to be more extreme, those that are risky, kind of, yeah. And then for this, we used the 10th percentile calculation, which assesses a specific environmental factor. And so anytime a point falls below the 10th percentile, we can classify it as a drought. So in order to do this, we take historical data over a long time period for a specific variable, and then we sort that from lowest to highest and calculate the 10th percentile. And then that value is the point that 10% of the data falls under. And then once the 10th percentile value is calculated, anytime the observed value falls below is considered a drought. The 10th percentile data is used because it does a better job of representing the rare events and conditions. And for this graph here, it's showing the red line is our threshold, and all the blue points are daily average stream flows in cubic feet per second. So total dissolved solids in the Mon have steadily decreased since 2009, and that can be directly affiliated with our voluntary discharge management program and the work that 3RQ does through our targeted water quality management and collaboration with coal companies. And then this is just really a proactive monitoring and intervention program. So during this most recent drought, the Mon had TDS levels that were below the Secondary Drinking Water Act standard of 500 milligrams per liter. The red dashed line in this graph is that EPA standard of secondary maximum containment level or contaminant level. And the blue line is the trend line for the TDS. And as you can see, it never passed that standard maximum. So we're doing pretty good. And then, of course, there's the Dunkard Creek fish kill in 2009, and really like the whole reason we have 3RQ. So the fish kill happened across 37 miles of the creek. And this is primarily due to the toxic algae bloom as a result of TDS. And it killed almost all of the aquatic life there. Drought conditions led to this event, including the increased TDS from mine drainage. This event led to further awareness and efforts of improved water quality. And ever since we've decreased TDS and increased the Mon's resistance to drought. So this graph here is just showing TDS levels and stream flow for Dunkard Creek since 2009. And then, so at 3RQ, we're really proud of all of our accomplishments. We have been able to identify causes. So most notably, WRI efforts have been crucial to TDS and water quality, and really understanding the high levels of TDS that contributed to the fish kill. We're also really proud of our collaborative monitoring that we do with different local groups, universities, government agencies, to create a more comprehensive view of the watershed. And then our restoration efforts and those of local coal companies through the voluntary discharge management plan, we were able to quickly decrease the TDS levels by 2010. And since then, the main stem has not exceeded that 500 milligrams per liter. And then lastly, we really work to educate and engage the public by sharing our findings and encouraging stewardship of local watersheds. And at 3RQ, we'll continue to monitor and work with groups to maintain those TDS levels. And then I included a link to the full story map if you're interested in all of the details of this. But yeah, I'd like to acknowledge Mel Schaefer, Rachel Spernak, Sarah Nelson, and Melissa O'Neill. And then of course our 3RQ sponsors, Colcom and the Foundation for Pennsylvania Watersheds. And I'll leave it to questions, I guess.
All right, thank you so much, Hannah, and all of our speakers today. You all did a great job. We do have a little time for some questions and answers, and it looks like that's already started in the chat. So I'm just gonna give you some highlights. So Luca and Aaron, do you have any recommendations to prevent or slow the upstream invasion of invasive fish? And I'm just going to read Erin's response from the chat. And she said, so currently there is a huge commercial fishing effort to reduce the number of adult carp in those lower pools where they are well established. We currently think that they are not able to recruit to their populations upstream because there is no real juvenile habitat in those middle pools. I believe that the continued removal of adults and potentially identifying juvenile habitat and removing them there as well would likely be very helpful in reducing spread upstream. Right, and then there's one more question in the chat currently, but feel free to keep typing those if you're still working on one. For Leslie and Citraka, what is the impact of significant levels of alkalinity at a testing site? Like what could happen with just that specific, I don't know, is it an element? Alkalinity. Tell me more.
I can let Citraka weigh in. Also, I'm not sure I totally understand the question, but what we found on the whole, the alkalinity, looking across the basin, we saw significant increases in alkalinity and or no observed trends. For that, I'm not sure I understand completely the question. So let me, if we have more time at the end at the questions, I can come back after I ponder a minute on that.
No, thank you for just elaborating and clarifying a little bit more. And while we wait to see if there are any other questions, I do want to just mention kind of what other sessions we have to look forward to in this series, as well as our other series. So next month in January, we are going to have a couple of presentations related to acid mine drainage to restored streams and critical materials recovery. So we hope that you'll join us for that. And then we also have our 3RQ series ongoing in 2 weeks. So we kind of offset that way. And they are going to be talking about collaboration through the Common Waters Initiative, which is new and very cool. I see we've got a hand raised. So is it Than, if you want to go ahead and unmute yourself?
Yeah. Hi. Thanks for the great presentations. It's Nathaniel's full name, Than, T-H-A-N is my nickname. It's really great work. I had a follow-up on the PDS and sulfate, story from the Mon. It's such a great story. And 2 questions. One is, I noticed there were some outliers. There was a few sites that bucked the major trend. So they did see increases in conductivity, for example, over time. And I'm wondering, is there something that explains that? Is there an obvious explanation for that, for those outliers you observed? And also, I would, love to follow up with you on the reverse osmosis treatment and the other sort of wins that you've seen over time, because the Guyandotte will now be going through a TMDL process for ionic pollution. I work with the West Virginia Rivers Coalition now. I was with USGS for 20 years. Now I'm with West Virginia Rivers, and we do see some an important agreement to move forward on restoration for the Guyandotte. So this is a really important opportunity. Sulfates are a big part of it, of course. And so lessons learned there would be great. So just to summarize, can you explain the outliers on conductivity trends that were interesting? And then 2, do you have thoughts for how the Guyandotte could benefit from the story of the Mon? And I guess this is to Leslie and to Hannah, or anybody who has thoughts on it.
Well, I can just kind of summarize. So our conductivity measurements were field measurements, and our TDS were from from our lab analysis. So that you might expect to see the same trends between the 2 parameters. So that might define why some of our sites for conductivity weren't tested as significant as to others. And also our significance level was an alpha of 0.1. So we're just showing our trends with that level. And we're only showing monotonic trends for not those non-parametric testing. So just kind of clarify in that way, that was our cutoff, whether or not we determined a trend or no trend. And I think I'll stop there maybe if Hannah
I
wanted.
could also clarify, what I saw is that some sites were going up, other sites were going down for conductivity. So I thought that was kind of unusual. Most of the sites were decreasing, as you'd expect from TDS going down, but there was at least one site in the main stem, and it was a real quick, I could follow up with you on this, but it's interesting that one site seemed to be doing something different, and I'm wondering, is there obviously some kind of explanation for that based on land use or other local effects? So, and I can also follow up with you, but that's the
And
question.
I'll follow up with you and share more of our results with you. Yes.
Hannah, did you have anything to add on to that?
I did not. Sorry, I didn't have anything to add. I feel like Leslie did a good job of answering all of that.
Yes.
And I can jump in just a little bit. So I know with the reverse osmosis system, whenever that went online in 2000 and I think 14, 15 maybe, we had actually already seen a very significant reduction in TDS on the MON as a result of total discharge management. So we worked with industry to essentially hold back their permitted discharges during low flow periods. So it was a very simple and effective approach versus putting it through a very expensive reverse osmosis system. So the reverse osmosis system went in because of the lawsuit settlement with Consol over the Dunkard Creek fish kill. But it essentially wasn't necessary because we already solved the issue. Not saying that reverse osmosis doesn't work. It's just sometimes there's other management opportunities that are more simple and straightforward that can solve the issue rather than just building a multi-million-dollar plant that has a lot of upkeep. And I'm not as familiar with the Guyandot to know if there's any other potential management solutions that might help during low flow periods, or if it's more of a constant flux, regardless of flow regimens.
Yeah, that's good insight. Thanks, Melissa.